Good afternoon, everyone. So today we're going to be talking all about controlling buffalo flies and fly lesions and what we have learned. My name is Fiona MacArthur and I'm a member of the ParaBoss extension team. For those of you who may be new to ParaBoss, it's a joint initiative of Australian Wool Innovation, Meat and Livestock Australia, the University of New England and Animal Health Australia. Shortly we'll be joined by today's presenter.
Good afternoon, everybody.
Yes.
Good afternoon, everybody. I want to do two things in this talk. First of all, I just want to do a quick run through of what we know about controlling buffalo flies and roughly what the recommendations are at the moment. And then in the second part of the talk, I want to talk about an MLA project that we've been involved in over a number of years, looked at the possibility of breeding for resistance as a tool to buffalo flies and to lesions as a tool in control programs. Where are we? Okay, so buffalo flies. Buffalo flies are now rated as the most costly health issue in the Northern Australian cattle industries, in fact, in all the cattle industries. And this is basically around two issues, the impact of the flies and the impact of the lesions. Now, buffalo flies are a nasty little critter. They're pretty much obligate parasites sitting on the back of cattle and just biting. They feed up to 20 to 40 times. And they're not like mosquitoes, which have nice fine mouth parts which stick into the capillaries. They have very rough mouth parts. What they do is create a little wound, and then suck up the blood. Now, this is highly irritating to the cattle. I'm just going to show a little bit of video that I want for those of you who may not be all that familiar with the effects of buffalo flies, and talk about a couple of things that I'll come back to later. So here's a cow being really heavily impacted by flies. You can see the clouds of flies down below her belly and above the back, and how they lift off her when she disturbs them and then come back onto the same animal. That head tossing that you've seen is classic buffalo fly response control, as is the tail switching and the kicking that we saw just a minute ago. So I'm going to come back to this in a minute. But that biting and irritation the flies could cause up to about 15 kilograms reduction in weight loss over the course of the buffalo fly season, and something like a reduction of half a litre of milk per day in dairy cattle. Now, the other major impact, sorry, let's stop that. The other major impact is of the lesions. And they don't actually cause that much economic loss, because most of the lesions occur around the neck and around the head, in places where they can actually be trimmed off skin fairly easily. However, where they do have an impact is in the sale yards, and where animals are regularly downgraded in terms of their value if they've got lesions. And this has become particularly a problem in our northern cattle industry. And this is because there's been a confusion between the wounds caused by lumpy skin disease, which of course we don't have in Australia, and lesions from buffalo flies. And you see in the bottom right-hand corner, there's a picture of an animal with lumpy skin disease, quite circular, sometimes a scarring in the middle or a hole developing there, but reasonably regular and lumps around the body. Whereas buffalo fly lesions are much more variable and much more irregular, and they really, and as I said, they're mainly around the eyes, as you can see here, and on the neck and flanks. You could perhaps mistake this one I'm circling up here, but for the most part, they are easy to distinguish. Now, buffalo flies are an introduced pest. They were introduced probably with buffalo from Timor in 1838, and they've slowly, or not slowly to start with, but more quickly later on, spread from Darwin, where they first came in, down around the northern parts of Australia, and then down the east coast quite quickly, until they got to here at Bundaberg. For some reason, they seemed to stop. This was about 1946, and that range was maintained until about 1974, and then they took off again, probably for three reasons: a series of wet years, the use of armatraz for ticks, which doesn't have any effect on buffalo flies, and because at that stage the requirement to treat animals for ticks before moving them was removed. Since then, they've moved down much further down, as far as, well, they've been recorded down at Maitland, and they regularly come up onto the New England tableland now from this coastal area where they can overwinter in wet summers. This area, they tend to survive around the coast, and then they spread out to this fawn area, possibly during when cattle move out, probably during wet summers. But the modelling also suggests that they're going to go further south, even with global warming, that the climate might get warm enough for them to become permanent fixtures in southeast and Western Australia, and in South Australia, if they actually get down there. So they're a growing pest, and I think there's a lot of people in central New South Wales that are now dealing with buffalo flies. And as Fiona suggested, we're just interested in seeing if we can find if they've gone any further south. So if you're down in those marginal areas, if you can let us know and chat, that would be great. Now, just a couple of quick things on biology of the buffalo fly. They're pretty much permanent residents on the backs or flanks or belly, depending on climatic conditions, of the cattle. And they only leave to lay their eggs in dung. They fly down when a cow defecates, lay their eggs, and then the larvae develop in the dung. The major this is where the major effect of them dying out during winter happens, when it becomes too cold and too dry for them, and they can't then complete their biology, and the population dies out. They then, what actually happens for their spread further west again, we're not really sure. They don't fly all that far. In most cases, if there's cattle there, they won't go more than 1 or 2 kilometres. And about the furthest reliable record we have of them spreading is infesting Magnetic Island from Townsville, and that's across water for about 7 k's. There's occasionally longer ones, longer distances registered, but they don't go that far. They probably spread more often on the backs of cattle with the cattle. Now, control of buffalo flies is an integrated approach, which could consist of all or some of the below: strategic chemical control. And by strategic, we commonly say that an economic threshold, the threshold where it's actually what you're going to lose is more than what it costs you to treat if you don't treat, is about 200 per animal, or 100 per side for beef cattle, and about 30 for dairy cattle. Sometimes people use buffalo fly traps, sometimes dung beetles, and sometimes resistant genotypes. And now this is historically been around the use of Bos indicus types or zebu types in the more northern areas. And they're more resistant than the more so-called British breeds in relation to buffalo flies. I'm also going, the major project that we've been involved in is looking at the selection within breeds for both lesions and for flies. And this has been done to some extent with lesions, but not much with flies. And I'll talk about some aspects of this in the later part of the talk. So so functionally, control is pretty much around chemicals. These other ones I'll talk about as well are helpful if you're in a position or in management situation, or an area where they could work for you. So the really important thing to remember about chemical control is there are a number of different chemical groups. And this is important in terms of managing resistance. The three main groups in buffalo fly control products are synthetic pyrethroids, and I've listed some of the major actives there, but they're all synthetic pyrethroids. And I've listed down below what products are available. So synthetic pyrethroids are available in ear tags, in pour-ons, in sprays and dips for buffalo fly control. Organophosphates, macrocyclic lactones, there's the so-called MLs or mectins, and they're pretty much only in ear tags or pour-ons. And then we have just a couple of combination products which have both OPs and SPs, but they're mainly sprays. And there are a couple of products which have repellents or things like DEET and dust, which are more registered for treatment of individual animals, perhaps some bulls or horses where they become infected. Now, I'm not going to talk much about this at the moment. I'm just going to point out there's a lot more information available for this in ParaBoss, and I'm just going to quickly show you where to find this. If you just follow this progression up here to from ParaBoss to FlyBoss, then over here to the tools on this top bar, down here to the products and animals, just filling these in. And then if you come down to buffalo flies, you can get a listing of a whole lot of information about application methods, about the different chemicals. But the really critical thing is to know these groups so that you can rotate them for resistance management. I'm going to talk about this a bit more later. The other place where you can find nearly everything you need to know about the products, including good pictures of the labels if you need them, is on the APVMA website. If you just search for PubChris, this website will come up and you can go in there and find your way through products from there. So they're good. These are good places, both on ParaBoss and on PubChris, to get a bit more information about chemical control. All of those methods were basically direct treatment of cattle. I'm just going to talk a little bit here about other methods which mean you don't functionally, or most of them do, mean you functionally don't need to get the cattle in to treat every 3 to 4 weeks. And of course, these, the most commonly used of these are back rubbers or back straps. You can buy them as mobile back rubbers, so you can move them around a little bit. And this is a more recent type of a design. These can be good where you can put them in places where cattle are going to frequent a lot, particularly if they're going to walk through gateways, for example, to water, or perhaps in areas under trees where they frequent. The animals will get used to knowing that they can get relief from buffalo flies from these, and they will actually most of them will learn to actually use them. There's a couple of good websites where you can that you will find quickly about back rubbers in Queensland for buying different designs of back rubbers, and there's a lot of really, really good information there. The other sort of automated ways that we can think of treatments are these spray races, where you run the animals through. These will require quite regular retreatment in most instances. And then I've got a couple of things that are used a little bit in the US. These automated misting devices, which usually deliver a fairly soft chemical, which isn't going to last very long, mainly for use on dairies where they're quite concerned about the use of chemicals. And they can be placed where the animals they're automated, there's a electronic eye that automatically sets off these misters and just delivers this as they walk in for milking or walk back out. The other one, I'm just putting up more for novelty use. This is registered in the US, and actually, interestingly enough, it was originally developed by a guy in Queensland, but then it went to the States. And this is using functionally a paintball gun shoot little paintballs of insecticide onto the backs of cattle. Now, there's a few issues about treatment here, and I'd be surprised if it ever gets registered in Australia. But you know, it's the sort of thing you realise was around when you were a kid, and you can sort of think that you might be able to get the kids out there doing a bit of treatment for you if you had this way. But as I said, there are some issues around how effective this is and around treatment, and it isn't registered for use in Australia. Chemical resistance. Now, this is one of the issues with all of our chemical controls for just about all of our parasites. As I said, the three main groups of chemicals that we have are synthetic pyrethroids, organophosphates, and the macrocyclic lactones. Resistance to synthetic pyrethroid, this is why it's important to know which group your product is in. Most synthetic pyrethroids, there's resistance to synthetic pyrethroids in pretty much all areas. The only good thing about this is there's actually a fitness disadvantage in the flies for resistance to pyrethroids, and if they haven't been used for a while, the flies will actually lose their resistance to some degree, and you'll actually get a pretty good effect from pyrethroids after you haven't used them for a while, when you've been using other chemicals. And I should actually make the point here, when we're talking resistance, in most instances, we're not talking that they don't work at all. We're talking they don't work as well, and that the period of protection will be significantly reduced depending on the level of resistance. Resistance to organophosphates also occurs. It's been out there for a long time, but it hasn't really become widespread. We've done some trials recently for other reasons, where we've been measuring around at sites around Queensland. We actually haven't found any OP resistance, so it's there and you need to manage it, but it's not nearly as widespread as synthetic pyrethroids. And for the MLs, there's no resistance that we know about. So important to know which group the products you're using are. A few rules for resistance management that I'm just going to flick through here. First is, as I said, know your chemical group so you can rotate them. And the point here is rotate chemical groups, not just between products. You need to know that the products you're going to rotate are in different groups. The second thing is don't treat when you don't have to. So if your management system allows it, don't do your first treatment, or don't put the tags in until you reach those economic thresholds. You know, as I said, in beef cattle, that's about 100 flies per side. Critically, follow the application instructions, correct dose, correct application methods, and you know, this is by far the main reason for breakdown in chemicals. Remove ear tags at the end of the life, as noted on the label. This is important, so you don't expose the flies to periods of low chemical, because below the effective levels, because that's what really selects for resistance quickly. And remember that the same actives are used for a number of the other parasites. And when you're treating those parasites, you're also affecting buffalo flies. So try and get your rotation systems up, taking into account the other things you're going to use. And as I said, critical. Read the label. We always say it, but just another reminder. And this is just the sort of resistance rotation strategy that people might use. If you're using tags, this is going to be the chemical group that you're going to use most often. And what you really want to try and do, if you have to do an early spring treatment and you want to delay until you actually have to tag the animals, you work out what tag you're going to use and then use a spray from a different group in that early treatment. And similarly, if you need a late treatment down here in autumn, winter, use a different chemical to which you've used in the tags, just to knock off the end before they go into winter. The other thing, as I said, ML porons also what you're going to use mainly for worms. So if you can time this treatment to get optimal effect against worms and buffalo flies, that's great. And where you're only using tags and you're not likely to use any sprays, you need to rotate your tags group across seasons. Don't use the same group in 2 seasons. And as I said, just try and work in with your treatments for other parasites to avoid selecting for resistance. Buffalo fly traps. These can work. Now, I'm not aware of anywhere you can buy buffalo fly traps in Australia anymore. I'm sure there's some fabricators out there that will make them for you. There is some plans for a buffalo fly trap that you can weld up at home on the web in the DAF Queensland site. So you can go in there and find some traps. How these work is that as the cattle walks through the trap into the shade, the flies don't like that shade, and they'll leave the backs of the cattle, and then they'll go to the light in these baffles at the side of the trap, or in the case of this one, which was the CSIRO trap, up into this cavity above the trap, where they'll get caught, can't feed, and they'll dry out and die. And then the ants do a pretty good job of removing them for you. These are really good, once again, where you can get good access and the cows are using them daily. This is the results of some work where Rudolph Urek and Peter Green, who some of you might know, looked at the Queensland trap in a beef cattle situation. These are the purple bars are the ones that didn't have the buffalo fly trap, and the purple ones where they did. And you can see that they definitely got a reduction in buffalo fly numbers, and particularly down here at the end of the season or before the season, it can keep your flies below an economic threshold, and it might save your treatment or something. So it's a great way you can get them in the area where you can get the cattle using them all the time. Bit more difficult in extensive rangeland situations. And as everywhere, I'll just put this picture in. You can't see it very well, but everything's bigger and better in the States. And there's some quite elaborate fly traps there now that have big vacuum systems where the flies get vacuumed off at the start and vacuumed off in the middle, and then vacuumed off at the end, just to try and get as many flies off as they can. As far as I know, these are not available in Australia, and they are pretty expensive, and you've still got that access issue. Dung beetles can have, in the lab, they have quite a significant effect on survival of larval buffalo flies, but they've had less effect in the field. For various reasons that we can discuss if you want to. So they can reduce populations, but they'll only ever be good as part of an integrated control program. And in that period when the flies are really going, they can just outbreed the dung beetles. However, having beetles brings a whole lot of other advantages, and so they're great to have in the system, particularly for nutrient recycling, and they also help control the other flies. They're very good against bush flies and also against Culicornis midges, which can bite cattle and can also carry blue tongue. If you are going to use dung, if you have got dung beetles, you need to be a bit careful with your buffalo fly treatments because some of them can have quite a toxic effect on the dung beetles, particularly the macrocyclic lactones and SPs, which are secreted mainly in the feces. OPs are less damaging because the OPs are secreted in the urine, and they have less effect in the dung beetles. But where the cows are still urinating on top of the dung, that can have an effect. There's a gradation in the MLs about which ones have the biggest effect against dung beetles. Doramectin probably have most effect, and moxidectin least within the MLs. The other point to note is that buffalo fly tags don't affect dung beetles just because they put the chemical on the outside rather than in where it's going to get excreted in the dung. Yeah. This last part I'm going to talk on some work that we've done over the last few years, basically aimed at breeding for resistance to flies and lesions. And I'm going to draw heavily on some PhD work done by Dr. Noman Nassim and Mohammed Cameron, and I'll introduce you to them at the end of the talk. Now, I've already said that we know that there's a difference in susceptibility to flies and lesions between Bos indicus types and between Bos taurus types, but we haven't really looked nearly as much at the potential for breeding within bloodlines or breeding within breeds. Now, the first point, and this is a really important point that I should make, is that lesions and flies in a breeding context have to be considered different traits. They're not the same trait. And by that I mean that if you breed for resistance to lesions, which we know can work over a period of time, and I'm going to talk more about this in a minute, that doesn't necessarily mean you're also going to get a difference in resistance, in numbers of flies. Okay, so you've got to think of them in a breeding sense as different traits. And just to illustrate that, I've got a graph here from some of our work. This first bar are cattle within the same herd with no lesions. This is a group with low lesion scores, and this is a group with high lesion scores. And you can see that there's no significant difference between these three groups in the number of flies that they had. And in fact, the ones with high lesion scores actually had not significantly different, but lower numbers of flies than the more lesion resistant groups. So just remember, think of them differently. So firstly, resistance to flies themselves. Now, most of the economic effect that we get from buffalo flies is due to the buffalo flies, that irritation that I showed you, and the removal of blood and reduction in weight gains. As I said before, lesions per se don't have a lot of economic effect, because we can trim them off in most instances. Now, we know, and you'll be aware of these cattle, talking about flies, not lesions, within herds, there are always this group of cattle that carry more flies, carry most of the flies, high fly burdens, flies, not lesions we're talking about, than a lot of other cattle in the herd. This is just a graph of one of our experimental herds, looking at the different cow numbers and the number of flies that they carried. And you can see that there's a group of cattle up here that carry much higher numbers of flies than the rest of the herd. Okay, so there's a small proportion of the herd that carry the majority of the flies. And we know from experiments where we've very accurately counted the number of flies on each of the animals, that this difference in the number of flies is heritable, and it's at least moderately heritable. And if we can count the number of flies accuracy, or measure the number of flies and get those heavily infested animals, the heritability is really quite high. Now, the difficulty comes in accurately estimating susceptibility, functionally in accurately counting the number of flies. Now, how can we measure the number of flies on different animals so that we can select the ones that have lots? Well, the first thing is just to directly count the number of flies. Now, this is difficult. It's really high labour. Most people won't want to do it, and they particularly won't want to do it more than once. However, if you do it a whole lot of times and actually count them, you probably get a pretty good measure of differences between animals. Now, the difficulty here is that buffalo flies are different to just about anything else that we measure to select for. So in body weight gains, if you weigh an animal and then you weigh it again in a couple of minutes later, you expect to get the same number. Even with worms, if you're breeding for worm parasites, you expect that if you take a fecal sample, then you go out and measure the worms, then go out and take another fecal sample, you'll pick out the ones that have got high worms, and the same animals will turn up in each time. With buffalo flies, it's quite totally different, because if you disturb those flies and they spread to other cattle, you can get a totally different count on that same animal. If you count it in a race, run it back round and count it again, and you get a quite different measure. So it's really difficult to get accurate measures. So I'm going to come back to how we might be able to get better fly count, direct fly counts in a minute. And the second thing that we might want to do to select for resistance to buffalo flies is to select on indicator characters. What characters make some animals more resistant than others? And ultimately, what we're looking for here is genomic markers. So the golden chalice, if you like, is being able to find genomic markers that identify the more resistant animals, so we can just take a blood sample, get it measured, and then come back on the basis of that genomic measure, work out which one's going to be successful and which ones are going to be resistant. But the catch-22 for that is that we need a good direct counting system, so that we can get good measures on lots of cattle to accurately identify these genomic markers. So we're back to direct fly counts again. Okay, so direct selection of buffalo fly counts. Now, unless you disturb them, as I said before, visual and photographic counts, or counts between observers, are highly correlated. Between observers, some might count higher than others, but generally the ranking, which is what you're interested in, the rankings of cattle are pretty much similar. So from that point of view, that's good news. And the high correlation, the counts, if you just take a count on a particular body area, like on one side or on the back, or even on the belly, the correlation between that count and the whole body counts in our work has been quite high. So you really only need, in most instances, to count a small area, as long as it's the same area on each of the animals. And greater number of times you can repeat those counts, the better estimate you're going to get. The catch-22 is that when we do this, functionally, the easy way of doing this is to do it in the yards. The downside about yards is that when you do this, the cattle are almost inevitably going to get disturbed, and they move between animals. So if we look at doing yard counts, we're going to get a correlation from a single count and an overall susceptibility of about 0.3 to 0.5. Whereas in paddock counts, as I said before, those flies, if they do get disturbed, they tend to go up in the air and come back down on the same animal. The counts are much more accurate, around about 0.6 to 0.8. So if we can get paddock counts, that's good. But this correlation, even with yard counts, is good enough that you're going to make some progress selecting on the basis of yard counts. It just won't be as good as paddock counts. And of course, you can, if you just be more careful and try not to disturb them as much, you'll probably be able to do a better job. Just going back to our photographic system, and as I said, the photographs work quite well. We've just been looking at some systems to make it easier to try and automate the measurement. And you can see we can visualize the flies on the animals in photographs quite well. And we can actually reduce it to this sort of an output. This is just along the back of an animal, which we can count in an automated system. So counting does lend itself, photographs and automated counting is quite a good possibility. Now I'm going to go to and I'll just go back to the other slide. Just going back to it. Of course, if we could do that in an automated system, once again, where the animals went through a race and got photographed automatically as they went to water along the back line, that has got real benefits in terms of potentially getting good measures of susceptibility. Okay, so coming to indirect selection, what characters could we use to identify the more resistant ones? The things that people have looked at before are things like colour and hair coat length and stuff, and they're associated at some level, but they're quite honestly not very reliable, and many studies have not found an association. We looked in here at the association between behaviour, and by behaviours we were looking at head tosses, ear flicks, tail flicks and foot stamps, and the correlation was actually quite good, even though we only counted the number of behaviours in 2-minute periods every week. And we still got quite high correlation, so it was giving, even for that level of counting, we were getting a good association. So what about if we could measure in a more automated basis? We could measure for a much longer period or continually. We would expect that the correlations might be even better. So we've been doing some studies at the potential of using accelerometer ear tags, which measure the head movements to characterise the amount of behaviour in those animals. And we did this by setting up the videos with the tagged animals in here, so we could match the amount of behaviours here with the tags here. We get an output here, which now we're sorting through all of that data that we've got, huge amount of data, trying to be able to use the patterns in these outputs to identify the differences in behaviour between animals. That's ongoing work. The second thing that we looked at was proteomic markers in the proteomes in the blood of animals, which might be different between resistant and susceptible animals. And two things came out with a number of markers. One was the blood coagulation pathways, and the animals that had high indicators for blood coagulation or clotting were more resistant to buffalo fly feeding. So we think what's going on here, that the animals with better clotting pathways, it's reducing the blood supply for the animals, and the animals go somewhere else because it's easy to eat in, easy for them to feed in. And the other group of them is in immune response identifiers. And here we think that this might be, we're not quite sure what's going on, other than the animals with higher general immune responses may be able to mount a response earlier and more strongly than more susceptible animals. And there's now a lot of interest in breeding for immune resilience or overall immune capacity, and it'll be interesting to see whether that might also have an effect on buffalo flies. So a couple of possibilities here. So this is in terms of, just to recap, for selecting directly against numbers of flies. Now, what about lesions? And I'll just flip through this because it's getting a bit late in the talk. Now, buffalo fly lesions, we've always considered that these lesions were due mainly to a nematode, Stephanofilaria, which is carried and spread by buffalo flies, and you can see the small stages here in a skin sample here. And these had an effect on lesion development, and indeed they are associated with lesions. However, working in southeast Queensland, we were never able to find these nematodes in cattle, even though there were lots of lesions. So we felt there might be something else going on here. Now, finding these lesions has always been difficult. It always had to be done by taking skin samples, and this is not very sensitive at all. So what Noman, the student working on this project, did was to develop a PCR means for measuring the presence of Stephanofilaria, which was much more sensitive than using skin samples. And then we looked at the presence of these nematodes in flies and in lesions in north, central and southern Queensland, and indeed in northern and central Queensland we found Stephanofilaria there pretty much all the time. But despite looking at thousands of flies in a number of places in southern Queensland, it reflected what we'd found before, that Stephanofilaria just didn't seem to be down here. So this suggested that other factors were involved in whether animals got lesions or not. So we then looked more closely at pathogenesis of lesions, other factors which are probably involved. We looked at three things. We looked at response to buffalo flies, and I'll come back to this again. We looked at Stephanofilaria in these lesions. We looked at bacteria that we found to be associated with these lesions, and we looked at allergic response in cattle. And what we found was, yes, Stephanofilaria there, and Staphylococcus was there as well in most of these lesions, and the same bacterial presence in the buffalo fly. So they were definitely spreading the bacteria as well. And we found that both of the bacteria, both the bacteria and Stephanofilaria, when we looked at the pathology of the lesions, they increased the pathology of the lesions. But what was more interesting was when we looked at the allergic response of the animals which got high lesions as compared to the ones which got low lesions. So we looked at the allergic response to buffalo fly antigens, the antigens injected by the buffalo flies. We did the standard allergy test, which any of you that have had allergy testing might have seen, intradermal skin injection with the buffalo fly antigens. Interestingly, we got a big wheel occurring at 24 hours, within 15 minutes pretty much. Then by 24 hours, we got this exudate forming around the lesion sites in some of the animals. When we looked at the size of the wheels and the skin thickness in animals which had high numbers of buffalo flies, the purple ones, as compared to the low lesion scores, we found that the animals with high lesions had much bigger immune responses to the antigens than did the more resistant ones. Now, this is good news in terms of breeding, because this says that there's a difference between animals in whether they actually develop lesions or not, that there's an animal response and that it's different between animals within a herd. And so in terms of breeding for lesion resistance, once again, we know that some cattle have high lesions year after year, and it's the same animals. Secondly, the rankings of these animals for lesion susceptibility are pretty repeatable within time. And furthermore, that the highest expression of the buffalo fly lesions was at the end of the season. And unlike flies, where they move around, once they've got a lesion, you'll get the same measure. So it's much easier to measure for or score than is flies directly. And we now have some evidence to show that this is inherited. And there are people who have been selecting against lesions over a number of years, and they will tell you that they've made progress in increasing resistance or decreasing susceptibility by doing that. So good news for that. The other thing to note is that now in Breedplan, there is a system for recording buffalo fly lesion scores. And so you can now, for those of you who are using Breedplan, you can now upload your lesion scores to Breedplan, and when there's enough records in there to give good estimates of breeding values, that will become available in Breedplan as well. So some good news backed by what we saw in our science in that project, and also some more indicators about how we might go about treating lesions. So that's all I'm going to say in this talk. Just a couple of very important acknowledgements, if I can.
Thanks, Peter. That was fantastic. I certainly learned a lot more about buffalo flies, so very informative and great to see where research is taking us and what solutions might be available for producers in the coming years. I have a couple of questions, Peter, that have already come through on the Q&A panel, but it's not too late for anyone in the audience who would like to ask Peter a question. Just pop it in the Q&A now. So the first question is, Peter, do you know of any AI tools that are in the making to use to count flies in real time or during a time window, maybe just a video or a photo that records the distance to avoid disturbing the animals?
There's a number of people looking at different methods of visualizing the flies and measuring from things like photographs. But the really big issue that we've got is how we actually get, as I think I said, how we actually get good things, good values to measure. It's all about being able to get a reliable number of flies on the animal to accurately identify which animals are resistant and which ones are not. So, using AI with the behaviour thing might work that way, and certainly, different ways of visualising the flies on the cattle might shortcut a little bit, but even just using ImageJ and Photoshop, we can do that in the lab already. So. There may be some work, but the really big area that we need is that automated photography, and then and using the sampling. What we actually measure, I guess, is a key element that needs to be taken into account as well.
Thanks, Peter. Next question: how much does buffalo fly affect cattle activity? For example, grazing or mating, drinking.
Yeah, I did have it mentioned to say that one of the explanations for the reductions in body weight that occurs, which is, as I said, up to about 15 k's in a buffalo fly season in a moderately affected flock, and that's been attributed to the behaviour, disturbance of animals grazing. It's also been suggested that that disturbance from the irritation can interfere with mating in some instances, and interfere with just making them much harder to measure. The other thing that I perhaps didn't mention in there is I mentioned about lesions and their effect on skins, and it's generally been considered that the lesions weren't much of an issue in terms of skin value, because you could trim off most of the lesions. But the other thing that has been shown in South America, now what's happened in South America is that they got hornfly, which is sort of the northern hemisphere version of buffalo fly, into Brazil in the 1960s, but into north of the Amazon in about 1980 into Brazil. And since then it's spread right down through South America into Argentina. And what they found in Argentina was that, as well, they don't get as much problem with lesions that we do, but what they found was there was a big difference in the tanned skin values in Argentina after buffalo fly spread down there. Now, so it's a thing called cockle, where the buffalo fly bites, you can't actually see that in the fresh skins, but when the skins are tanned, it leaves these small pinprick faults in the skins, which devalues the skin. And probably a lot of the reduction in skin values that is just routinely applied to values for skins in the Northern Hemisphere is actually, and is largely attributed to horns and to ticks, is also largely due to buffalo fly feeding. So that's another economic effect that perhaps we haven't recognised. And we've never had the opportunity like they did in Argentina, where they had before hornfly and then after hornfly.
Thanks, Peter. I've got a couple more questions just before we finish off. Do you have advice on when it's financially viable to treat buffalo fly?
Well, the rule has always been that the economic threshold, and that is that the cost of treatment, or the cost of treatment is going to be less than letting the buffalo fly run, is about 100 flies per side, and as I said, about 30 flies per animal on dairy cattle. Now, obviously, all sorts of things come into that. You know, the cost of your product, the cost of you, definitely the cost of being able to muster, and so, and also there's welfare considerations. So, you know, often you know, treatments. It's often said that treatments should start to be applied once the cattle are starting to show visible distress from the flies. But you know, trying to get those sorts of measures, it's difficult work. It's really very difficult to get that. But that's the number: 200 flies or 100 flies per side for beef cattle is suggested.
Thanks, Peter. Next question. Is there a resistance test kit available to test if we have when we have a problem in our herd?
No, there's not a routine test for that available. It's usually, you know, we can do it in the lab. It's basically a treated surface test, but there's no one doing it routinely that I'm aware of. I may not be correct in that, but as far as I know, but it's not terribly difficult for those two products anyway.
Thanks. Final question. What rotation strategy would you recommend if using OP tags are banned under the current APVMA diaznon reconsideration?
It's a huge issue, and it's something I probably should have mentioned. I think the short answer is it's going to be a big issue, and I think that we should be doing as much as we can to try and see that doesn't happen. And I think there's, you know, I think there's my personal view, and there's other factors around markets involved here as well. But as I talked about with the effects on buffalo flies, I think the issues with organophosphates from tags is much less than from other methods of treatment. And I think we should just be trying to win that fight before we have to deal with it. Other than that, we can just hope that there's a different chemical group somewhere in the development process. And other than that, with tags, we're left with two groups: SPs and the ML tags. There's one ML tag on the market.
Thanks, Peter. One last quick question that's come through, and I think, yes, we do have time. Could you please elaborate on what exactly it is in beef and dairy cattle that acts as an attractant for buffalo fly when we're coming back into the warmer months after a cooler spell? Is it the animal scent or a particular blood type?
Well, look, the stuff with it depends on what level you're talking about, I'm afraid, and by two levels. With most parasites, including buffalo fly, they've got to find an animal first, and very often with parasites, that attraction comes from a distance. And it might be things like just urine and fecal smells from a whole mob of cattle, and carbon dioxide is often attractive to biting flies. Then we've got the effect of whether there's an odor effect up close. That is the reason that explains why some animals have flies and other animals don't. And there's been a bit of work done in this space, but the evidence that there's a particular smell associated with why animals have more buffalo flies is not particularly in my view, other people might disagree, is not particularly compelling. Our information and some work that's been done on buffalo fly, on vaccines to hornfly, would suggest that the animals that have more flies are the ones that are easier for the buffalo flies to get a blood meal from. In other words, the blood stays, it's easier for the flies to feed on the more susceptible animals than on the more resistant animals. So they go, have a bite, think probably this is a bit hard, and they tend to accumulate on the animals where it's much easier to feed.
Great, thank you very much, Peter. That brings us to the end of our presentation. We've run a little
Can I just say one other thing I should have said about the resistance thing? Of course, the other thing that I should have said in terms of resistance is that you don't. It's not just tags you're dealing with, and if we've got less actives in tags, it would be really good to intersperse that with a backliner or a spray or a dip with a different chemical group, just to break that up a little bit. Sorry, Fiona.
Oh, not at all. Thank you, Peter. Great advice. And thank you to the audience. What a wonderful selection of questions you've put in for Peter this afternoon. That was really informative and interactive Q&A time. So thank you, Peter, so much for joining us. It's been an absolute privilege to have you online sharing your expertise with us.
Thank you, everybody.
Thanks, everyone. That's it from us here this month from ParaBoss. We'll see you again next month. Bye for now.